This show me details Chapter 15 Special Senses, specifically the eye/vision. As stated in your text, 70% of the body's sensory receptors are in the eyes. So you see that a lot of neuronal connections and also the processing in the cerebral cortex, remember this all happens in the occipital lobe, is dedicated to vision and visual processing. Looking at some of the accessory structures of the eye, we look first at this image where we can see the sclera, which is the white part of the eye. In the middle we see the iris, which is the colored portion, and the iris is, remember, a muscle that is in charge of controlling the size of the pupil. So the black part is the pupil. Remember, the pupil is not a solid structure. It is a literal hole that allows light to go into the eye. So there's nothing in that specific area. It's covered by the outer portion, which is called the corneum, which we'll look at in a minute. But we also, remember, have our eyelids. Okay, and we also have our eyebrows. These are all considered to be accessory structures of the eye. They are important for protecting the eye, keeping it moist, and also keeping things from falling into the eyes, such as our eyebrows as well. Now we also, remember, have this area over here, which we'll talk about in a little bit, that has a drainage system that allows fluid to flow, as you can see in this image, down and across the eye, and then down into our nasal cavity. In this particular image, we can see up front we have a protective membrane, a protective layer called the cornea that goes in front of the eye, but on top of the cornea is a structure called the conjunctiva, and you all might have heard of conjunctivitis. I spelled it wrong, conjunctiva. It is in charge of protecting the eye and protecting things from going back into the body. So as you can see, this membrane covers all the way around to our eyelid on both sides, so that if something were to try to come in, it would be protected and there would not be able to go further back into the eye. Which makes me think of someone who wears contacts, such as myself, and you have your contact that sits over the front of your eye. Sometimes your contact can slip and go backwards. Rest assured, it will not end up in your body because there is this protective layer of the conjunctiva that prevents it from going further back. In this image, you can also see the muscles here and here that are in charge of opening and closing the eyelids. You also see some of the muscles that attach to the extraocular muscles that attach to the eyeball that are in charge of turning and moving the eyes. Remember that this muscle here and here was one that you had to name. This is the orbicularis oculi muscle. So back to this image, this is the lacrimal system. Remember that the word lacrimal means tears. So we have a lacrimal gland that sits up here kind of in the lateral superior aspect of the eye, and it has these little canals, these little ducts that will drain down the tears onto the surface of the eye, and the tears will then flow across the eye towards the medial side. We have a little hole called a punctum right here that allows the tears to drain into the larger, what is called the lacrimal sac, and from there, through the nasolacrimal duct, it will flow down into the nasal cavity. So this is something that we've all experienced before, maybe when you have an emotionally upsetting time where you might have a runny nose because you have lots of fluid that's flowing down from your eyes into your nose. Consequently, also, when we're ill and this sac becomes inflamed and becomes blocked, we will have flow of the tears out onto our face, which you've all experienced both in emotional upsetness and in sickness. In your text, if you want to look over at figure number 15.3, I'll give you a second to find that if you have your book, or pause the video and go look at it. You will see that we have 6 extraocular muscles that are attached to the eyeball. We have the lateral rectus, the medial rectus, the superior rectus, the inferior oblique, and the superior oblique muscles. Now remember that the innervation for these cranial nerves, and I'm not going to require that you guys know specifically which one is what, but remember that they are cranial nerves 3, 4, and 6, okay, which is the oculomotor, the trochlear, and the abducens nerve. Each one of these muscles that is listed here has a specific action, and the first 4 are relatively easy to figure out because this one's going to move the eyeball laterally, this one's going to move the eyeball medially, this one's going to be superior, and this one's going to be inferior, and this one also has a slight medial. Both of these actually have a slight medial turn as well, whereas the oblique muscles, as you see, inferior is going to elevate the eye and turn it laterally. So superior and lateral, and this one's going to be inferior and lateral. So these 6 muscles collectively move our eyes. Remember that we have a lot of nerve input for 6 very small muscles, meaning that this area of our body, the eyeballs, has the finest motor control of all of our muscles. So they're the most precise muscles in the human body. So there's some imbalances that are related to these outer structures of the eye and some of the conditions related externally, such as the stye, which is an inflammation of the sebaceous glands that surround, usually in the eyelids. So sebaceous. And with the stye, if anyone's ever seen it, there seems to be an outpouching, so you see a large kind of growth that is usually present on the eyelids. Conjunctivitis is a condition where the conjunctiva becomes inflamed, reddened, irritated, usually scratchy. So this can be either pink eye, which is listed, which is a bacterial or virus infection, typically highly contagious, or it could just be maybe an abrasion that causes redness in the eye, if you ever had something that scratched your eye, scratched your cornea. Diplopia is a condition where, in literal terms, this is double vision. Okay, double vision. And this occurs when the 2 eyes, the muscles are not coordinated, so they're not seeing, they're not focusing on the same exact thing. So therefore 2 images are seen instead of one. And many things can cause diplopia, so it could be paralysis or just weakness of the muscles, or it could be something more serious, such as a neurological disorder. Now strabismus, the last condition here, aka cross-eyedness, can either be medial, which is what we typically consider to be cross-eyed, or it could be lateral, where both eyes are turning laterally. This is typically a congenital condition, so something that you're born with, and it's a weakness of the eye. So with this, the eyes alternate focusing on objects, meaning only one typically controllable eye is used, so the person does not see double vision. So when this happens, if you have 2 eyes and you have your optic nerves coming into your brain, but only one eye is really transmitting and the brain is only detecting this one eye, then the brain begins to disregard input from the deviant eye, which can lead to functional blindness if this person is not treated. Now, for the internal structures, this is what you guys are going to see when you do your dissection, so make sure you pay close attention to this part. Okay, the first thing to note is that the eyeball itself has three general layers. So we see these three layers right here. Okay, so I'm just going to put 1, 2, 3. Okay, the outer layer, which is called the fibrous layer, is made up of the sclera. I'll write it over here. Okay, the sclera. So the sclera is the white portion of the eye. It's very durable. You guys are going to see this when you do your dissections. And it's also made up of the transparent cornea, which is up front. Okay, so really there's two structures that make up the fibrous layer. The second layer is called the vascular layer. Okay, the vascular layer. So you see here that it's red. So up front, we're going to talk about some muscles that are important. Remember, muscles are highly vascular, but then this layer in the back, okay, which is kind of folded down here, is called the choroid. Okay, the choroid layer. And it's going to be the one that contains the blood vessels, hence the word vascular. So up front here, we see these two structures. Okay, one is going to be called, move this here. So this is the iris here. So you see the iris from a lateral view, and you can see the pupil here in the middle, which is, remember, the hole. And then this other structure is called the ciliary body. And we'll talk about a few of its functions here in a second. Now this inner yellow layer, this is the neural layer. So hence, we will see that this is where the neurons are located. Specifically, this layer is called the retina. So you guys have probably heard of the term retina. And this is going to be where the photoreceptors, make sure you know what this word means, photoreceptors are located in the eye. And we'll learn that there's two different types of photoreceptors. There are rods and cones. Now I'm zooming in here on this more anterior part of the eye. So remember, we have the cornea up front here. This right here, this is a chamber, and it's called the anterior segment. So it's a space, and it's filled with the fluid called aqueous humor. Remember that humor is another word for liquid. So aqueous humor means that it's a watery type of liquid that fills the eye. So we're going to see in a little bit that the ciliary bodies are responsible for creating the aqueous humor. So now that we know that there's an anterior segment, there must be a posterior segment. So where do you think the posterior segment's going to be? So the posterior segment is going to be back here, okay, behind the lens. So we have the anterior segment up front and the posterior segment in the back. The dividing point for these two segments is the lens. So this is the lens. So what does the lens do functionally? Why do we have a lens? Remember, the lens is what is going to help to focus the light, okay, towards the retina. So it's going to be in charge of focusing, changing our focal point. So if we're looking at something up close, and then we change our focal point and look at something far away, in an instant, it's able to shift our focal point. And so if you do this right now, if you hold your hand up in front of your face right now, everyone do it, and look at your hand, notice that behind your hand, everything appears blurry. Now, if you shift your focal point and look at something behind your hand, what happens to your hand? It becomes blurry. So this is an example of how we shift our focal point. And the term for this is called accommodation. So when the lens is changing shape to focus, it's called accommodation. So now, looking at this picture in front of you here, do you see how the lens will change shape? Can you see those little structures that are attached to the sides of the lens? Okay, so you should be looking here and here. And these structures are called the ciliary zonules. So they're very similar to like a ligament type structure. They attach from the ciliary body to the lens, and they can pull on the lens to change its shape. So the ciliary body here is really the structure that will contract to change the shape of the lens. So we have that surrounding the eye. Remember, this is a frontal section, really. Actually, it wouldn't be frontal. This would be a sagittal section, and it's a circular shape structure, so it goes all the way around the lens and completely encircles the lens. Okay, 360 degrees. Okay, so let's do a little review real quick. Can you remember what the name of this part was? And do you remember what we called this part? And furthermore, what was this part filled with? What was the name of the fluid? Okay, then we have this one. And this one. Okay, number 5. And number 6. Number 7. And number 8. So if you need to, go ahead and pause this and see if you can name these 8 structures, because I'm going to go over the answers right now. Okay, so remember, number 1 was the cornea. Number 2 is the anterior segment that is filled with the aqueous humor. Number 3 is the iris. Number 4 is the pupil. Number 5 is the lens. Number 6 are the ciliary zonules. Number 7 is the ciliary body. And number 8 is the posterior segment. Let's now shift our focus to the back side of the eye. So the back side of the eye, remember we said this area is called the posterior segment. It is filled with the fluid called vitreous humor. So vitreous humor has a higher viscosity than the aqueous humor, and you guys will see when you do your dissection, you can literally hold it in your hand. It looks kind of like jello. You can kind of shake it and it wiggles, and it fills the back side of the eye, and its purpose is to keep the retina in place on the back side of the eye. Another thing that you guys are going to see is that the retina is a very delicate tissue, and just one swipe of your finger against the retina will cause it to move and change shape. So this vitreous humor is very important because it keeps the retina plastered against the posterior wall of the eye. Okay, the next structure back here is important. This is the optic nerve. So this is the transmission point where all of the retinal receptors are bringing in their input, and they're converging on the optic nerve that will then transmit this signal back through the optic chiasma, which remember is the crossing, through the optic tract, which will then bring the signal into the occipital lobes. We have a structure here where there is no retinal tissue. So if you see the yellow located all the way here and here, notice that there is no yellow here. This is called the optic disc, and it's also called the blind spot of the eye. So there's a little area of the eye that does not have photoreceptors, and therefore does not receive light signals. The next part here, this is called the macula. And right in the center of the macula is a very important structure called the fovea centralis. Okay, the fovea centralis. Fovea centralis functionally is important because this is where we have our best visual acuity. Meaning, if we can focus the light on that area, it's where we have our best vision. This is probably a good time to bring up the 2 types of photoreceptors found in the eye. So we have rods and we have cones, which we mentioned before. Rods are specific for black and white. So black, white, or shades of gray. Insert joke, 50 Shades of Grey. Just kidding. Cones are responsible for detecting color. So when we are using our rods, compared to when we are using our cones, think about when do you see black and white and gray versus color. Think about light and dark, like when you're in vision at night versus day vision. So cones and color are seen better in bright light, whereas the rods are detecting black and white and gray really in dark. Now we can also see those colors in the daylight too, but think about when it's dark out, you don't see bright colors. You see black, white, shades of gray. So the reason I bring this up is because the fovea centralis is only made up of cones. Only made up of cones. So there's not enough light for us to focus and see, like if we're trying to read a book and it's pitch black. We obviously can't see it. We don't use our cones we don't use our cones at that point. We're just using our rods because it's dark. So that fovea centralis only for cones. We use this when we have bright light available to us. Rods contain a pigment called rhodopsin. So it's a specific pigment that builds when we are in dim or we're in darkness. That pigment will build and it will allow us to slowly see better. So you guys have probably experienced this if you've gone out into the dark and you notice that your vision improves after a period of minutes or so. So the rhodopsin is important. It's a purple, kind of dark purple pigment. And the reason I bring this up is because it's been a final exam question before, so make sure you know it. This is figure 15.5, and it details two important muscles that will be in charge of changing the shape of the iris, thus increasing or decreasing the size of the pupil. So on the right, we see this is the process of dilation. So this muscle is called the dilator pupillae, and it is sympathetic. So when you think about sympathetic control of the eye, think about fight or flight. Usually you're in danger, you're exercising, you're embarrassed. It causes your pupils to dilate to bring in more light to assess the situation going on around you. Whereas on the other side, parasympathetic stimulation will stimulate the muscle called the sphincter pupillae to constrict. And so we see that they have different mechanisms of action. The dilation over here pulls the muscle out, thus increasing the size of the pupil, whereas the sphincter muscle rotates in a circular fashion to close or constrict the pupil. What this image is it is just a kind of a review of the circulation of the aqueous humor. So remember that it's produced over here in the ciliary body, and it's produced by filtration from your capillaries. So the fluid just doesn't arise out of nowhere. The fluid has to come from somewhere. So the fluid is taken from the bloodstream, from the capillaries. So it's kind of similar to blood plasma. It doesn't have all the things floating around in it. It's just the fluid. And the fluid is then circulated out and around. So you see it actually travels through the pupil and then comes out into the anterior segment. Okay, and actually there's two subdivisions of the anterior segment. You have the anterior chamber and the posterior chamber. So those are just subsegments, or subchambers, I should say, of that segment that's all lying in front of the lens. So then we see the little hole down here. Used to be called the Canal of Schlemm, which I thought was kind of a fun word to say, but your textbook, as it's been changing a lot of the structure names back to anatomical terms instead of people's names, canal of Schlemm, is now called the scleral venous sinus, which up until this edition of the book I've never actually heard of, because I learned it, the canal of Schlemm. Just like when you guys saw in the book the node of Ranvier. Do you guys remember where that was? Where was the node of Ranvier? So if you're thinking neuron, you're thinking correctly. It was the place where there is no myelin. And if you remember now, your book calls it the myelin sheath gap. So the new text has gone away from really using a lot of people's names and more towards what is the actual anatomical structure that's going on in this particular part. So just a little side note. Have you guys heard of glaucoma? So glaucoma is a fairly common condition in which the pressure in the eye is increased. So it's increased intraocular pressure. Typically it's because there's some kind of blockage that prevents the fluids from draining. And the danger with glaucoma is that the pressure can build to a point where it can push against the retina and the optic nerve, which can lead to blindness. So the danger with it is there's really no early signs of it. You only have signs or symptoms when the glaucoma is pretty far set. So it is dangerous for this reason. Any of you who go to the eye doctor and you sit in front of that machine that blows that puff of air onto your eyeball, if you go, you know exactly what I'm talking about, because no matter how well you prepare and know that that air is about to hit your eye, I still have a violent reaction when it hits my eyeball. So the point of that machine is that it blows the air onto the eye really quickly, and then it measures how fast the tissue bounces back. And if it bounces back really quickly or faster than normal, it can be deduced that the pressure inside the eye is higher than normal. Okay, the last concept we're going to cover is refraction. So if you guys remember, refraction is the way that light travels. So when light travels through, think about light in our eyeball. It's going to have to go through the cornea, into the pupil, past the lens. So you see that here. Cornea, lens, all the way. And so we see in an emmetropic eye, this is a normal eye, the focal point is exactly here on the retina. When we have problems with our eyes, so the first problem is called myopia. Okay, myopia. Myopia is nearsightedness. Okay, nearsightedness. So with nearsightedness, the eyeball is too long. So as you see here, if the eyeball is too long, the focal point falls in front of the retina. Therefore, when it hits the retina, there's almost divergent rays. So you see that it will cause blurriness. So with myopia or nearsightedness, the correction is that there is a concave lens that will initially diverge the rays of light so that they converge on a longer focal point to the back of the eye. And oppositely, we see a condition where the eyeball is too short. Okay, too short. So with a hyperopic or hyperopia, the eyeball is too short, therefore the focal point lies behind the eye, meaning in the same fashion as before, multiple rays hitting the retina in different points causes blurriness. So the corrected the correction for it is to use a convex lens so that it converges faster so that the focal point lies right on the retina there. So some of you out there might also have heard of, or you might have, what's called an astigmatism. An astigmatism is different than these other two because it is a misshapen eyeball to the point where the cornea or the lens has abnormal curvatures, And so therefore, special lenses have to be used. Okay, special lenses have to be used for these people to be able to see properly. As you know, people with astigmatism, your frames and your contacts are going to cost more money, because if you just have normal hyperopia or myopia, you can just go to the optometrist and they can give you a standard contact because your cornea is the same shape as everyone else's cornea. But with astigmatism, yours has to be specially made because you have weirdly shaped eyeballs. Sorry to say it, but it's true. This concludes our section in looking at the eyeball. Please make sure you prepare well for the dissection. If you need to go on YouTube, you can see videos of eyeball dissections, or if you want to search, Google search images, you're more than welcome to do so as well. So the rest of the chapter details the other senses, which are smell, taste, hearing, and equilibrium.